So today we are going to study about the techniques which are used to study the molecular markers. So you know what are molecular markers? So molecular markers are like variations in the DNA sequences by which you can differentiate between the two individuals or between the two populations or even two different species. But there are some specific techniques which are used to study the molecular markers. One technique is the random amplified polymorphic DNA technique. So what happened in this particular technique? In this particular technique, you are using a single primer. Like in my next slide, you can see this is the primer which you are using, and the same primer is working as a forward primer, and the same primer is working as a reverse primer. So you are using a single primer. Usually in the PCR, we are using a two primers, forward and the reverse primer. But in the RAPD, you are using the single primer, which is working as both forward and the reverse primer. Now, whatever the region is present in between these forward and in this particular sequences, that is going to be amplified. It could be within the range of 0.2 base pair to 5 kb, for which the PCR is pretty much sensitive. So this is the fundamental rule of the RAPD. You are using a single primer. A single primer is working as both forward and the reverse primer, and the region in between these two sequences are going to be amplified. For example, this region is going to be amplified. So you can see here the RFPD was developed by Welch and McHenk along with Williams in 1990. It was pronounced as the rapid because it was pretty fast. It is based on the PCR assay and it does not require any prior sequencing of DNA. You are not doing any sequencing, you are just doing the PCR by using a single set of primer. This procedure uses short primer sequences of 8 to 12 base pair that randomly amplifies the region of DNA. This primer serves as both forward and the reverse primer. This reaction proceeds when a single primer anneals to the complementary strand of the DNA template. This amplification of segment of DNA depends on the positions complementary to the primer sequences. Okay, so only the middle region is going to be amplified. So you can see here the fragments obtained from RAPD are in between 0.2 to 5 kb, and because this is a PCR technique, you can see the bands by doing the agrogel electrophoresis after staining with ethidium bromide, or you can even do the native page. So this particular technique is a binary technique. Either the band is present. If the band is present, then the score is 1. If the band is absent, the score is 0. So if there is any mutation in the primer binding site, you will not get any amplified DNA band. So you will not get any PCR product. If any mutation occurs in the primer binding site, then no PCR product will be produced. So in this case, you can say if a primer binding site is present, then if suppose primer is binding to this particular allele, and if the individual is capital A, then there will be the presence of band. If the individual is capital A or small a, because capital A is present, again you will get the presence of band. But if the individual is small a, because this is created because of any deletion, duplication, like this, then in that case, primer will not bind to this site and there will be no amplification. So what you can see here, you can easily see here that this particular technique is dominant in nature. it cannot differentiate between the homozygote and heterozygote. If the individual is homozygote, you will get the band. If the individual is heterozygote, you will get a band. So you cannot differentiate between this, and that is the reason this particular technique is dominant in nature. And again, presence of band, you can say if the band is present, you can give the score of 1. If the band is absent, you can give the score of 0. So it's a binary data you are going to obtain. Okay. Okay, so what are the applications? Distinct pattern of amplification you can see in the different samples, because in some individuals it will bind to, let's say, this particular region is going to be 0.2 base pair, but in some other individuals it could be 0.5 base pair or something like that. So you can differentiate them. RAPD is applicable for the mapping of genome, analyzing linkage and individual specific genotyping. And how come it is like that? For example, you are studying two markers, let's say marker 1 and marker 2. Okay, and you are getting the office springs, 16 office springs like this, either 1 1 or 0 0, means either the marker 1 is present, marker 2 is also present, or either the marker 1 is absent or 2 is also absent. In the 4 office spring, you are getting marker 1 is present, but marker 0 is absent, marker 2 is absent, or marker 1 is absent or 0, marker 2 is present. So these are the recombinants. For marker 1 and 2, these are the recombinants, because in one it is present, another it is absent. In this case, these are non-recombinant. So can you calculate the recombination frequency? So recombination frequency is basically number of recombinants and total number of offspring. So number of recombinants, how many? 4. Total number is 16 plus 4. So 4 by 20 means 0.2 centimorgan. It means marker 1 and marker 2 is present in this population. It should be like 0.2 centimorgan apart. Like this you can do for the other markers also, and you can create the genomic map. You can also identify the linkage. If suppose you are always getting the marker 1 and marker 2, always you all the 20 offsprings are showing 1 1 and 0 0, it means they are linked. Okay, so that's how you can this technique is applicable. These are dominant in nature because you cannot differentiate between the capital A and capital A small a. It's homozygotes and heterozygotes. You cannot differentiate, and this is the reason this technique is dominant in nature. It is strictly laboratory dependent, so it requires sensitivity. Demerits: poor reproducibility, field faint bands, and band scoring is also a problem, because sometimes what happened, you are getting a band like this. It looks like two band, but it could be one band also. So you cannot say whether this is both are present or this is a single band. So scoring is a different, because the scoring is done on the basis of presence or absence. If presence, you will give 1, absence, you will give 0. So sometimes the scoring is a concern in this technology. So here you can see in this particular uh, slide, this is the DNA template. You are using the primer. The same primer is binding to the working as a forward and the reverse primer, and the middle region is going to be amplified. And that's how you are getting multiple bands. See here, same primer is binding to this side and this side, so you will get this amplified fragment. The side of the PCR fragment is going to be this much. In this case, it is going to be smaller. So like this, you will get the multiple bands in the RFLP technology. Now coming to the RFLP. So what happened in the RFLP? You extract the DNA, you digest it with the restriction enzyme. So once you digest it with the restriction enzyme, you will get multiple bands. Now you will separate these bands from the agar gel. You do the southern blotting. Then you do the hybridization of these fragments with a particular probe. And then you can detect the probes by the autoradiography if they are radioactivity labeled. If these probes are fluorescence labeled, you can use the non-radioactive system as well. So what happened here? If you see here in this case, this particular technique is co-dominant in nature. For example, if suppose in one, uh, if the individual is capital A, and the restriction fragment is going to recognize this sequence, you will get the restriction enzyme will cut this particular fragment. Why it will cut? Because it is going to recognize this sequence. Okay, so you will get the smaller fragment. Okay, if the individual is, let's say, capital A, small a, then in the case of A, it will give you a smaller fragment because the restriction enzyme will recognize this particular sequence. In the case of a small a, because it is not recognizing that particular sequence, then in that case, because of any deletion or duplication, the restriction enzymes is not recognizing this sequence, so you will not get any fragment, cut fragment. Okay, so you will get a larger fragment, which is this one, and you will get the smaller fragment, which is this one. Okay, so in the case of AA, you will get two fragments. One is larger, one is smaller. In the case of small a, you will get a this particular thing is not going to be recognized by the restriction enzyme. Then you will get the larger fragment. So how you will see here, if you see, so in the case of capital A, you will get this fragment. In the case of capital A, small a, you will get this fragment. And in the case of a small a, you will get this fragment. So you can easily differentiate between the capital A and capital A small a. And that is the reason this technology is so dominant in nature. Because it can differentiate between the homozygous and heterogeneous. Okay. So it was the first method used for the analysis of DNA in the various fields in the forensic sciences, like that. It is a hybridization based technique because whatever the fragments you are getting, you are hybridizing it with the probes. It was invented by the Alex Jeffreys, an English scientist, in 1984 during his research in genetic diseases. It uses the restriction endonuclease enzymes, which are cutting at a specific site, and the length of the distinct fragments is determined by using blotting. Now you can do it by the sequencing also. RFLP markers are locus specific and are co-dominant in nature due to the nature of the restriction endonuclease used. Like in this example, you can also see if the individual is homozygote, you will get, because it is going to be recognized by the restriction enzyme, so it will cut this particular sequences. You will get a smaller fragment. In case of capital A, small a, you will get two fragments, and in case of the small a, because this site is mutated, restriction enzyme will not recognize this sequence. You will get a no fragment, so one larger fragment. If you have a 200 base pair sequence fragment, then restriction enzyme is not going to cut it. like that. So you will get a larger fragment. RFLP was one of the first technique applied for genetic fingerprinting and profiling. You can identify the inherited diseases. If suppose the disease is caused is recessive in nature, let's say, and it is caused because of the small a, then you can identify if the individual is small a by the RFL. If the individual is capital A small a, is a carrier of the disease, you can also identify. Means the heterozygous conditions also you can identify. So if there is any mutation or deletion, substitution, insertion in the site recognized by the restriction enzyme, then you will the fragment is not going to be cut by the restriction enzyme. So you will get a different fragment size. These alterations in genome can be detected by analyzing fragment of variable length digested with the restriction endonuclease enzyme. So the demerit is this: restriction enzymes are costly in nature. Sometimes they are contaminated. Sometimes you are using two restriction enzymes at a time. So the conditions of the restriction enzyme should match. So it's a laborious and a tedious technology, and it requires a relatively larger DNA sample. Why larger? Because you are having a full DNA and then you are fragmenting it. Now the third technology is AFLP. If we talk about the AFLP, you can see here in this particular technique, you are using the rare cutter, one restriction enzyme, another is the frequent cutter. So whenever you are using the combination of restriction enzymes, it should be one should be rare cutter, which is recognizing 6 base pair. One should be frequent cutter, which is recognizing the 4 base pair sequence. For example, ECO-R1 is recognizing the 6 base pair and MSC1 is recognizing the 4 base pair. You cannot randomly use any restriction enzymes. You need to use a restriction enzyme, combination of enzymes which could work together. One should be the rare, one should be the frequent cutter. So if once these, for example, E4R1 is recognizing the GAAT site, so you will get the overhangs. Now these overhangs, here you can see the overhangs of AA. These overhangs needs to be filled by the adapter molecule. So you have the adapter molecule that will fill these overhang sequences. Similarly, in case of the MSC1 also, you are getting the overhang of AT. So your adapter should have the TA sequence in it. Once this is there, you have the adapter, you have the restriction enzymes, and you have the fragment. Now you know whatever the fragment you want to study. So you design the primers against the adapter and this restriction enzyme, and then you add a particular sequence. So this particular sequence is going, suppose if it is A, so in the complementary strand, if it is T, this will bind and you will get a fragment. Amplification is going to be there. But again, there might be many fragments which have the T. So this is not very specific. So to increase the specificity, you are adding two more base pairs. Sometimes 5 base pairs people are adding. So in that case, if you are adding AC, so in the complementary strand, if it is T and G, then there will be the amplification. So this is to enhance the specificity of the reaction. But again, this particular AFLP is dominant in nature. What does it mean? It cannot differentiate between the capital A, capital A, capital A, small a, because this is a PCR based technique. And even if one A is present, the amplification is going to be there and you will see the band. Okay. So in that case, you cannot identify. You cannot differentiate. If suppose capital A, small a is there, you will get the band and you will not get the band. Then how will you differentiate in between capital A, where you will get the band? Because you are not getting multiple bands here. So this is the technique which is again dominant in nature. It cannot differentiate between homozygotes and heterozygotes. It cannot differentiate it. So Javier and Bose invented the AFLP uh, technology in 1993. So RFLP in 1984, RAPD is 1990, and AFLP in 1993. It was first developed by Cuisine in 1990. It's a PCR-based technique. It includes both PCR and RFLP. So the first step is DNA extraction. Then you digest it with the restriction enzymes. One should be the frequent cutter, one should be the rare cutter. Then you add the adapter sequences to fill the overhangs. Then you amplify them by the PCR, and then you analyze them in the gel electrophoresis or the native phase followed by the autolytic grafting. Now, what are the applications? AFLP has its ability for rapid generation of marker fragments for any organism without prior sequencing. Means sequencing is not required in both the cases, RAPD and AFLP. Okay. Also, the fragment size is smaller. The smaller DNA fragment size is required in the case of AFLP. Is largely used for the crop improvement programs, parentage and genomic interpretation of various crop species. Demerits require larger DNA samples and require purification. So that's all of today's time.